Greetings to Professor Dr. Wan Lian. We are from Group 17 and today we are discussing an article, Genome Sequence of the Modernized Variety KITAAKEX, written by Jane et al. 2019. The overview for our presentation based on the article will start with the introduction, methodology, result and discussion, conclusion, and will be ended with comments from our team members regarding the article. This article is a study about Oryza sativa, which is an important food source for many people and is also used as a model for studying other plants. Based on the previous study, different types of rice have been sequenced, but their use in functional genomics research is limited due to long growing periods and low transformation efficiencies. Hence, to overcome this limitation, researchers turned to a variety called KITAAKEX, which has a fast life cycle, is easy to transform, and can be propagated efficiently. This rice variety has become a preferred model for studying not only for rice but also other similar plants. In the study, researchers sequenced and analyzed the genome of KITAAKEX, which refers to a KITAAKE plant carrying the XA21 immune receptor gene.
However, the incomplete genome sequence of KITAAKE will block its use in the rice research. For example, Cascade9 will guide the RNAs cannot be accurately designed for KITAAKE without its complete genomes. Hence, a high-quality genome sequence of KITAAKEX was assembled and compared to Nipombare and Soso Jensen 97 genomes and identify its genomic variation, making KITAAKEX a model for infection disease study as the XA21 gene confers resistance.
The objective of this study is to report the de novo genome sequencing and analysis of a specific rice variety called Oryza sativa subspecies japonica variety KITAAKEX, where it is aimed to provide a high-quality reference genome for KITAAKEX, which is a model organism for functional genomic studies in monocotyledonous species. The first method to conduct this study is to plant the seeds of KITAAKE and other rice varieties. The KITAAKE seeds were provided by Dr. Hiroyuki Ito and other rice seeds were provided by Dr. John E. Leach and the USDA Del Bumpers National Rice Research Center. The seeds were germinated and grown in a greenhouse in Davis, California. The plants were planted in pots and each pot contained 3 plants. The plants were grown in a greenhouse where conditions include the specific temperatures and light intensity during the day and night periods. The heading date when the first panicle appeared was recorded for each plant, and the experiment was also repeated during the winter season.
The other method that are used in the paper is the construction of the phylogenetic tree that involves the calculation of IBS distometry using genotype of all rice accession, including the 3010 accession of the 3K Rice Genome Project, and some additional node accession that are fetched from the SNP databases and related genomic data. The IBS distometry was then used to construct a phylogenetic tree by the unweighted neighbor joining method for the genome sequencing and assembly method, where a high molecular weight DNA from a young leaf of KITAAKEX was isolated and used in the sequencing. Next, annotation of the protein coding genes were obtained through a high throughput RNA sequence analysis of library from a diverse rice tissue and gene production using various methods such as protein-based homology search, followed by annotation with SwissProt, TREMBL, and Interprobe databases, and the pathway analysis with the best matched aquatic protein in the KEGG databases.
Now we go to genome synchrony. We use SynMap to identify collinearity block using homologous CDS pair. After that, we use CellCost to visualize collinearity block. Now we go to repeat annotation. Repeat masker and blaster were used to obtain the fraction of transposable element and repeated sequence in assembly. Reconciliation of masker repeats was carried out using custom Perl script and formatted in GFF35. Results with score lower than the family-specific gathering threshold were removed. Only the highest score will be kept. Analysis of genomic fraction. To identify SNPs and indels, we use show SNP option with parameter negative CLRTH. We use the show code option of MOHD with parameter negative TRHCL to identify gap region PAVs above 80 base pair. Inverted alignment block with more than 98% identity was used to identify inversion.
For the next method, bacterial artificial chromosome library is constructed where the BSC clones are stored until needed and the restriction enzymes used are HEN3 and the BSD-YI. And finally, method to estimate the genome size where the histogram of 24 mer frequencies was created using Illumina fragment library. and the camera histogram indicates a peak value at a specific frequency is equivalent to the average coverage of 24 months on the genome, and the peak value was taken and computed the total bases in the Illumina library, and lastly the total bases was divided by the coverage, which then provided an estimate of the genome size.
The result obtained from this study is that a variety of rice, KITAAKEX, was identified to flower much earlier than other sequence rice varieties. Under long day conditions, KITAAKEX starts flowering at around 54 days after germination, while other varieties like Nippon Bare and 9311 flower much later, ranging from 79 to 134 days after germination. To understand the relationship between KITAAKEX and other rice varieties, a phylogenetic analysis was performed using data from a study that examined the population structure and diversity of 3010 rice accessions, and it was revealed that KITAAKEX and Nippon Bare are closely related and belong to the same subpopulation within the larger population of rice varieties.
The result for the genome sequencing and assembly shown that the KITAAKEX genome was sequenced using a combination of SR and LR sequencing in a high quality of de novo assembly that contained 377.6 MB, consisting of 33 scaffold and with a total of 99.67% coverage of assembled bases in the chromosome that can be seen in the figure here. And the assembly for the sequence completeness using BLAT result show that 98.94% of the genes completely aligned, 0.75% partially aligned, and 0.31% were not detected to the KITAAKEX assembly. And the overall error rate in the BAC clones is 0.09%, indicating that the high quality of the assembly.
Now we go to genome annotation. In this paper, they predict 35,594 protein coding genes in KITAAKEX genome. The predicted protein coding regions are distributed unevenly across each chromosome, and the gene density is highest at the end of chromosome. KITAAKEX genome then compared to complete rice genome to assess the quality of the annotation.
The result for the genomic variations between KITAAKEX and the other rice varieties can be detected by the comparison. And in this study, they compared the KITAAKEX genome to the Nippon Bare and Zensha 97 genomes, including the 4 aspects that are SNPs, indels, PAVs, and eversions using OMAR. Okay, based on the table shown here, they identified the variations among the rice genomes by looking at the SNPs and indels. Let's look at the figure 2b shown here, especially 2b and 2c that is circled in yellow. It is also shows the comparison in terms of SNPs and indels, but in a visual way. As we can see in the figure 2b that is circled in blue, the genomic variations between the KITAAKEX and the Nippon Bare are highly concentrated in some genomic regions, but in the figure 2c that is circled in pink, the variations between the KITAAKEX and the Zensha 97 are spread evenly through the genome.
For discussion, Nippon Bare genome was annotated as a high level, but it got a long life cycle. The availability of a high genome and annotation for KITAAKEX will be useful for associating trait of interest with genetic variation because it's got short in life cycle.
For the 219 small variations that has been identified between the KITAAKEX and the KITAAKE genomes, it has been discussed that these small variations may have resulted from the somatic mutations that arose from tissue culture and regeneration, and also from the spontaneous mutations. And mutations can be detected by aligning rates of the KITAAKEX mutants to the Nippon Bare reference genome, and it is resulted that the KITAAKEX rates covered on Nippon Bare is in quite high coverage, that is on average of 97% of the Nippon Bare. This proves that the KITAAKEX sequence is useful as direct reference genome. For example, it is used as a reference genome for the KITAAKE due to it has only small variations, which is only 219 SNPs and indels, and also to detect mutations. In conclusion, the de novo assembly of the KITAAKEX genome serves as a useful reference genome for the model rice variety KITAAKE, and it will further facilitate more research into the genomic studies of rice and other species, such as including their biological traits or genetic improvement.